So first I have to explain what we talked about last class. And I see now why I made the mistake. I've had a little bit of a sleepy past week, but just from the orientation. So this is actually a great thing to review, and it will be a great final exam question. So this will prepare you for one such question. So remember, when we're talking in photosynthesis, we drew the chloroplast, and then we mentioned how inside the chloroplast there are these stacks, right? And what are those stacks called? The thylakoids, right? There are these stacks, and that's where photosynthesis is occurring. And inside the chloroplast, this is called the stroma. It's kind of equivalent to the cytosol, right? And this is inside our thylakoid, and that's called the thylakoid space, right? And then photosynthesis is occurring in these photosystems that are embedded in the cell membrane of the thylakoid, and the electrons are passed down here. And as the electrons are passed down here, the drop in their energy at each fall is used to power the movement of hydrogen, right? And that hydrogen is going from the stroma into the thylakoid space. So here, our thylakoid space represents that enclosed space where we can build up a high concentration of hydrogen. So if we're building up a high concentration, what's the pH here? High or low? It's going to be low, right? You'll get more acidic inside here. And then, as we discussed, that electrochemical gradient can be used to synthesize the production of ATP. And that always occurs because there's going to be our ATP synthase. I should draw it like the turbine it is. That ATP synthase here, and the hydrogens are going to flow outside here from region of high concentration to low concentration, and that is used to power the synthesis of ATP. So our ATP is going to be produced in the stroma. We take our hydrogen from the stroma, we pump it into the thylakoid, they diffuse down their concentration gradient, and the ATP will be synthesized in the stroma as well. Well, in the mitochondria, I didn't put up a picture of that, but I will in a moment. In the mitochondria, remember there is there's an outer membrane here, and then there's an inner membrane, and that inner membrane is really folded to increase the space on which oxidative phosphorylation can occur. And between these two membranes, this is called the intermembrane space. And in this case, that is now going to be our enclosed space, as you'll see in a moment. Inside the mitochondria itself, it's called the matrix. And so the matrix is the or the same thing as the stroma as cytoplasm. And in oxidative phosphorylation, hydrogens are going to be pumped from the matrix, similar here, into the intermembrane space, which is acting as our enclosed space. So our hydrogens are going to build up inside here. So now the low pH, the acidic zone will be in here, right? It's equivalent of inside here. So these two spaces are the equivalent of each other in this model. And these two spaces are the equivalent of each other. And now our ATP synthase will be embedded on this membrane. And the hydrogens are flowing from high concentration down the concentration gradient. Oops, and I should draw it with its big ball of ending. And that ATP will be synthesized in the matrix. So our matrix is the same thing as our stroma, and our enclosed space is the thylakoid space in chloroplast, and it's the intermembrane space in oxidative phosphorylation. And then our proteins are embedded on those membranes, and in both cases the hydrogens flow down their concentration gradient, and the ATP is ultimately synthesized in the matrix or the stroma. So you can think of a variety of good exam questions here. I could ask you, which one is low or which is high pH, for example. I can imagine a multiple choice question like, which are the two regions in the chloroplast and the mitochondria that have low pH? Is it the thylakoid and the intermembrane, the stroma and the matrix, et cetera, right? And the correct answer would be intermembrane and thylakoid are getting a low pH. Right? So something like that would be a good multiple choice question. We don't have any long answer on the final because this would be a nice thing to draw as well. It's all multiple choice. Okay? So I just did a few PowerPoints, just put things together that we've seen before. So this is one of the pictures from chapter 10 showing that, and here's our stroma. The hydrogens are being pumped from inside the stroma into the thylakoid space. And remember that we have to build up our hydrogen concentration in a contained space. So the thylakoid space is that contained space in photosynthesis. And there's not very many pictures of this in chapter 9, which is why I messed up the other day. But so here, the hydrogens are being pumped from the matrix into the intermembrane space. The intermembrane spaces are enclosed space, and then they will flow down their concentration gradient, and ATP will be synthesized inside the matrix. So this was the picture that kind of describes it, but describes the situation. So you show here in your mitochondria, your outer and inner membrane, and how the ATP synthase and electron transport chain are occurring on this membrane. So in both cases, our enclosed spaces are intermembrane space or thylakoid space, and those are the regions to which hydrogens are being pumped from the matrix or the stroma. Okay, so this is the good one to relate them. Okay, any questions about that? Okay, let's get back to chapter 9. So this is where we ended off, and so remember we drew the electron transport chain as a series of 3 steps, and then our hydrogens are always being pumped from the matrix into the intermembrane space. That's the diagram. You should relabel those. That was a mistake I made. So we went from the matrix into the intermembrane space. And remember, NADH starts at the top of the staircase, right? And it's carrying 2 electrons. And its electrons get passed down through 3 protein complexes that are associated with 3 proton pumps. So there are actually pumps here. And NADH will pass its electrons through 3 pumps. 2, and then 3. 3 pumps. And where does FADH2 start? It starts at the second one, right? FADH2 bypasses the first pump, so it's gonna pass its electrons in here, and it's only going to be using 2 proton pumps. So I want you to remember more what you need to know is more within the context of what I have on the board, because I mentioned I don't want you to memorize the names of all of these proteins associated with the proton complexes that are going to be receiving and passing on those electrons. but I have missed something here, which is remember that those electrons are being passed like a hot potato from person to person, or people going, or the analogy could be people going down an escalator, and if the guy at the bottom doesn't get off, or the guy at the end doesn't pass on his potatoes, they're going to get filled up. And it's got to be, in the oxidative phosphorylation, it's ultimately passed to oxygen and makes water. And that makes sense because oxygen is so electronegative. So that allows the chain to continue going. But notice, because this will come up in a few moments, this chain would actually back up if oxygen wasn't present. And that's what happens in fermentation. So, such as when you exercise too much, as we'll talk about in a moment, if you deplete your cells of oxygen, what actually happens is all these electron carriers get filled up. And there's no one to pass their electrons to. So the whole system stops, and you stop making ATP through chemiosmosis. That's what happens when you don't have oxygen here. We'll talk about that in a moment. Fermentation will occur. Either lactic acid or alcohol fermentation. So one of the other things I wanted to mention is that one of the questions has been how many ATPs are we going to be able to generate for each hydrogen that is passed through here. And roughly for each of the two electrons, for each of the two electrons, we're going to pass 2 protons, which makes sense. Because each have a negative charge, they're going to be associated with the movement of 2 protons. So 2 electrons will cause 2 hydrogens to go into here. So each NADH carries 2 electrons, right? So each NADH will be able to pump 2 hydrogens at each of these steps. Will be associated with the movement of 2 hydrogen at each of these steps. And then there's the question of how many hydrogens have to pump through for each ATP. How many ATPs do you get for each pair of hydrogens? And the answer is about 1 to 1.5. I think I said that correctly. I think that's correct. I will come back to this in a moment. So how is this ultimately going to happen? It's going to happen because of this protein which we've described a few times now, ATP synthase. And ATP synthase uses the exergonic flow of hydrogen to drive the phosphorylation of ATP. So this, the entire process, as we talked about, this specifically is electron transport chain. But the entire process of mixing this plus chemiosmosis is called oxidative phosphorylation. And it is called oxidative phosphorylation because in the end, what we're doing is phosphorylating ATP. And it's an oxidation reaction, and we are here, here an oxidation reaction is being used to create this hydrogen gradient, which will ultimately be involved in the phosphorylation of ADP to make ATP. And that's where the word oxidative phosphorylation comes in. So it's combining this entire process, oxidation process, with the phosphorylation of ATP in chemiosmosis. And as we've now seen at the beginning of lecture, this occurs because of this amazing enzyme, and this is a much better picture of it. It shows you here how the hydrogen enters like a funnel